Alright, class. So we did mention that action potential follows a action potential. Why is this science thingy here? Follows all or none law. What do we mean by that? So let's focus on the picture. The dendrites receive the information, right? So the red arrows, they receive and send the information towards the soma. Now, the soma integrates all the information. How it integrates, we will look at it later. But for now, just know that all the information, all the incoming information are being added up or integrated within the cell body or soma. And once threshold is reached, so inside it's -70, right? If it reaches, so as the sensory information comes in, if it reaches -55, then an action potential will be triggered at the axon hillock. And that follows all or none law, meaning no matter whether you have a super strong stimulus or a weak stimulus, whether you have 1 stimulus, 10 stimuli, or 100 stimuli, doesn't matter. If threshold is reached, meaning if it reaches -55, the axon will shoot an action potential. I usually compare this as killing someone. In today's political situation, it's probably not the best analogy, but let's say I feel threatened and I want to shoot at someone. Now, no matter whether I pull the trigger slowly or fast, if I pull the trigger, the bullet will go out, it will fire. So it doesn't really depend on whether I'm pulling the trigger softly or fast. Does that make sense? So that's the point. There is nothing called, so if I pull the trigger really slow, will the bullet come out slower? No. Once I pull the trigger, the bullet will come out. So that's the point. No matter how strong the signals are, no matter how many signals are there, if it reaches threshold, then an action potential will be generated, and that does not depend on the stimulus or the stimuli. Okay. So this is another review slide. Let's read through the slide and make sure we understand everything. So when we are at rest, a neuron is considered polarized, meaning there is separation of charge. And at that state, the inside is obviously more negative as compared to outside. When the membrane potential inside the cell increases, or we say it becomes less negative and more positive, this state is called depolarization, meaning the inside becomes less negative, more positive, and that typically takes place by entry of positive ion like sodium, calcium, potassium, etc. A return back to resting membrane potential is called repolarization, and when the membrane potential inside the cell decreases more or becomes further negative, this is called hyperpolarization, and that is usually done when positive ions like potassium leave or negative ions like chloride enter the cell. All right. And the picture on the right hand side is showing how you can actually record resting membrane potential by inserting electrode within the cell. As you insert electrode inside the membrane or in the intracellular space and record, you will see that the inside of the cell measures negative as compared to as compared to outside, and that is resting membrane potential. In this, on the monitor, we do see that the resting membrane potential of the neuron is set to minus 70 millivolts. Okay, ion gatings in axons. So, starting from the axon hillock all the way down, we have voltage gated channels, right? There are sodium voltage gated channels and potassium voltage gated channels. Now let's talk a little bit more about these channels. Potassium, if you think of potassium, as we mentioned before, has two types of channels. We have leak channels, right? And the leak channels are everywhere and they are always open, and potassium trickles out through the leak channels. It's like keeping the tap slightly open as water drips down. So we have this dripping of potassium through the leak channels. They are always open. Then there are voltage gated potassium channels, and when they open, potassium rushes out of the cell to the extracellular environment. Remember, the voltage gated potassium channels open at positive 30 millivolts. Then we have the sodium voltage gated channels, which are closed at rest, but they open at threshold. So at negative 55, these channels open, letting more sodium inside the cell, resulting in depolarization of the cell. We also have some sodium leak channels in just specific neurons. Not every neuron has sodium leak channels. But again, like the potassium leak channels, the sodium leak channels are also always open, and a little bit of sodium trickles out. All right. Looking at voltage gated sodium channels, we just finished talking about this, so I'm not spending time on this. Now this is the graph showing action potential. Where in the cell, where in a neuron is action potential generated? Let's draw. This is the neuron. Action potential is generated here at the axon hillock, right here. This is where the action potential is generated, at axon hillock, and the action potential travels all the way down the axon to the axon terminal. Okay? All right, so let's look at the graph. Resting membrane potential is negative 70, as we talked about. Negative 55 represents threshold. Then on the x-axis, we see time in milliseconds, and on the y-axis, we have membrane potential in millivolts. Now, there are a few phases of action. Well, there are a few things which are differently color-coded. Moving from resting membrane potential to threshold, meaning moving from negative 70 to negative 55, this is not action potential. This is called graded potential. What is this? Graded potential. We will talk about graded potential in depth later in this video. Not in this video, in another video. So this is graded potential. Remember, this is not action potential. And graded potential take place on the dendrites and soma or cell body. That's where graded potential take place. Remember, the action potential starts at the axon hillock and travel down the axon. Now, during this graded potential, what happens? Which channels are open and which channels are closed? Really important. You will get few questions on your exam just based on this information. So we have sodium potassium pump. Then we have sodium leak channels, potassium leak channels. We have sodium voltage gated channels. We have potassium voltage gated channels, and we have sodium ligand gated channels, right? So there are 5 types of channels which are open when the cell is moving from resting membrane potential to threshold. Well, at this point, we have the pumps open and the leak channels open. The rest are closed. Okay. All right. So once threshold is reached, nothing can stop. Nothing stops. There will be an action potential for sure. So when it reaches threshold, obviously there will be an action potential. And during that, if it would be nice if I can change color. Sometimes it does, sometimes it doesn't. I don't know why. Anyway, so the green line represents depolarization. So I'll just put the green line here. It's annoying. The green line represents depolarization. I'll put depol. And the membrane depolarizes until it hits positive 30. Do you see this point? It is depolarizing. Now, what is happening during depolarization? Which gates are open? Which gates are closed? Well, we have sodium-potassium pump. We have sodium leak channels. We have potassium leak channels. We have sodium voltage gated channels. We have potassium voltage gated channels. And we have sodium ligand gated channels. During depolarization, listen carefully. During the process of depolarization, just give me a minute. Where did I keep that? Anyway, I don't find it. Anyway, during depolarization, we have these open, since they are always open, sodium potassium pump, leak channels, potassium leak channels, and also the voltage gated sodium channels are open in this case. Okay. The voltage gated sodium channels are open in this case. All right, and then these are closed. All right, actually there is one little thing that I would like to change, and that is during graded potential, the ligand gated sodium channels are open. So this is open. So only the voltage gated channels are closed at that point. Okay. All right. So we understand which channels are open during graded potential, which channels are open during depolarization. Now let's take a look at repolarization. So the light blue line represents repolarization, and during repolarization we have, let's write these down, sodium potassium pump, sodium leak channel, potassium leak channel, sodium voltage gated channel, potassium voltage gated channel, sodium ligand gated channels. Okay, so during repolarization we have the sodium voltage gated channels closed. So remember, the sodium voltage gated channels close at here. So at positive 30, the sodium voltage gated channels close. We have the potassium voltage gated channels open at this point. And also the leak channels are open, and the sodium potassium pumps are open. And the sodium ligand gated channels are closed as well. That's repolarization. When repolarization brings back to, oh, actually I'll stop here and then start with a new video.